Hello and welcome to Learn Bioinformatics from the Scratch. You are watching part 4 and today we will discuss what is BLAST and how to use it. I'm Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. If you're watching part 4 and if you haven't watched 1, 2 and 3 parts, I would suggest first to watch those 3 parts. Why? Because in part first we have covered about some sequence and some sequence retrieval stuffs. In part 2 we have discussed the basic concept and how alignment is working. In part 3 we have discussed the concept of homology. Now today is a kind of practical demonstration that what is BLAST and how and why we will use it. So let's begin. BLAST actually stands for Basic Local Alignment Search Tool because it works on local strategy of alignment we discussed in part 2. It's perform alignment or searching of different sequences based on alignment. So because of its tool or the way it's working, it is called tool. So basically, so the name is coined from these kind of words: basic alignment, local alignment search tool. Okay, when to use? Before going into the practical demonstration, we will discuss these 5 points like why and when we will use BLAST. So first of all, if you are interested to search larger biological databases or large biological databases like GeneBank, UniProt, non-redundant databases, or so many others against your query sequence. What is query sequence? The sequence which you have will be called a query sequence. So if you have only one sequence and you want to find out how many other sequences contains homology to your query sequence, you can use BLAST. To find related genes or proteins across different species. If you want to know that what other genes and what other proteins will be related to my query sequence. So that's why you can use BLAST. 3. If the function of a gene or a protein is unknown. In this case, let's suppose you have your gene or you have your amino acid sequence of a protein, but the function is unknown. Then BLAST can identify similar sequences with known functions and it will provide you a clue like what kind of function or what kind of role your sequence will perform or it's expected to perform. Number 4. For example, you have performed PCR and you send your PCR product for sequencing and now the company sent you back the product sequence information. Then in that scenario, we also use BLAST to find out whether my PCR product is the correct one or not. We take that sequence received from the company, we BLAST it. If it hits majorly for the gene we took experiment or we amplified it, then it means our PCR product is correct and it is safe for to be used for in further experiments for protein expression and others. Number 5. For example, you have a data set. I would not say data set. I mean, for example, you have your gene or a protein sequence. Now you want to generate a data set of some sequences related to your gene or protein. And next, you are planning for some phylogenetic tree or some phylogenetic analysis. Then, in that case, we will use BLAST to to find out or to collect the related or homologs sequences. So let's perform BLAST and to see how BLAST is working. Well, I have opened this browser. Now, if you don't know the correct URL, just type BLAST. Okay, let me type this way. Google BLAST basic local alignment search tool. Click here. You will see this kind of window: nucleotide blast, protein blast, and this is translated nucleotide blast, and this is protein translated or reverse translation back to the nucleotide. So far we are interested in nucleotide blast and protein blast. Okay, if you remember in part 1, we have retrieved a sequence of NDM1 from a database. So we will use the same sequence which is still here on my desktop. This was the sequence we retrieved in part first, and we will use the same sequence in this blast. Okay, as our sequence is a nucleotide sequence or a gene sequence, therefore we will click here. But if your sequence is an amino acid sequence or a protein sequence, then you will click here. So once you click here, you will have this kind of window, this one. It will say enter query sequence. I told you in the start that your sequence is a query sequence. This is a job title. For example, this is NDM1, so you will write NDM1 whatever. This is a database. For example, if you want to search this database, then you can select all these databases. For this is for reference sequences, and this is for reference genomes. This is for nucleotide collection. Normally people are using this one, and it is also present by default. Okay, now if you are interested to perform blast again a certain specialized organism, then you can enter here the name or add organism. Otherwise, leave it blank. This other things, these are optional, so you can leave it blank. And program selection, if you are interested to gather highly similar sequences, then you use this one. Or if you are interested to gather some dissimilar sequences, not highly similar sequences, then you click here. But if you are interested somewhat in between, not so much related or not so much distant from each other, then click this one. But in this case, it's okay. We will go with this one. Okay. Now you have the sequence already downloaded or retrieved in part first. You can just copy the whole with the name, or you can copy this one. That's okay. Control C. Now paste it here, and let's say I'm naming it NDM1. Okay, and click blast. I'm going to pause this session not to waste your time. So let's see. Okay, we have our result page. You can see this is the ID of this blast search. Which program was used? Blast N stands for nucleotide. Which database we use? The NT or the nucleotide redundant database. And this was our query sequence. We didn't any description, no. What was the molecular type? Of course we use nucleotide, so it was the blast recognizes itself that it was a DNA. And the query length, it means how many base pairs was contained by your sequence you subjected to blast. So that was 729 bases. Okay, you can have your results in four different tabs: descriptions, graphical summary, alignments, and taxonomy. So in description you can see this page. It will tell you like Klebsiella pneumoniae, this strain, gene name is NDM1, complete CDS, coding sequences, CDS stands for coding sequence. And it says it gives you the scientific name of the organism from which this sequence belongs, and it is 100% matching, and the score is this one 1347, and the e-value is 0, and its accession number in the database is this one. The second one is actually the vector T blah NDM1. It's also complete sequence, but it's a vector, so the vector cannot be named from any specific organism, so that's why they call it a vector. The third one is another bacteria, the fourth one is another, and you can see again Klebsiella pneumoniae, but another strain L5151. Here the strain is different, and you can see the query coverage or the sequence, the score information. It's okay. Now what is the difference between the graphical summary? When you click it, the graphical summary stands that these sequences are comparing to or similar to your sequence, and the color shows, you can see all red. It means the red means that the score is greater than 200. If some of the sequences the score is below 200, it will you will see this color, this and this. So it also gives you a proper interpretation that what sequences are homologs to my sequence and what are not homologs. So what are the homologs? For that, you have to watch part 3 of this series. Okay. Now, if you want to see the alignments of individual genes, so you can also click here, or you can also click on the graphical summary. Let's say I want to see the alignment of this gene or maybe this gene with my query sequence. Click here. So once you click here, you can click here alignment. So you can see here the query is our sequence and the subject is the one picked from the database. These lines, the vertical lines, actually shows the convergence or the conserved regions, and I cannot see, yes, here you can see here a gap in your query sequence, or I would say the indel operations. If you don't know the indel operations, then please watch the second lecture of this series. Okay, so this was how you can interpret the blast results. Now this is a taxonomy. If you want to see what bacteria's hit in this blast result or sequences from what bacteria you got hits. So the number of hits total 102 and 101, sorry, total 102, and from, let's say, from Klebsiella we have 56 sequences, from Enterobacter we have 2 sequences, and likewise you can also have this kind of information. Now, for example, you are interested to draw the phylogenetic analysis or to perform some evolutionary analysis in next stage, then what will you do? You will need to download this data. So how to download this data? That is simple. Let me show you. Okay, but before to download this data, sometime you will see duplicate sequences. For example, you want to keep one sequence from each organism. Now you can see Klebsiella pneumoniae, one sequence is here, one is here. Okay, one is here. Although they are different strains, but based on your analysis, sometime you are interested to keep only one sequence from Klebsiella pneumoniae. You don't want to include all the sequences. In case you want to include all the sequences, then the game is very straightforward. Just download. But if you want to exclude some sequences, then what will you do? For example, you want to keep this one, but you want to exclude this one. So this time you will have some manual operation. Just uncheck this one, just uncheck this one, just uncheck this one. These all are the Klebsiella pneumoniae and see like this. Okay, once your data set is clear or you are now in a position to download, then how can you download your data set? Click here, and from here you will have a download. Okay, here on this screen, if you want to select or to increase or decrease some columns, then you can do. For example, you don't want to see the maximum score column, just click here. You can see here, from here the maximum score column is disappeared. Now you can only see total score. Let's say you are also not interested in this column, so you can see. Okay, now we are going to download these results. So here you have some options. If you want these results in the CSV, so the CSV will only give you the hit table. Hit table means only those amino acids or bases which are hitting or which are aligning perfectly. But most of the time, if you are a researcher, then you will be interested in the FASTA format. But here, FASTA format offers you two types of sequences. Complete sequences, it means without alignment, and FASTA sequences with alignment. So normally I ask my students to go for the complete sequences without alignment, because I intend two things for them. So I will do the same for you. Why to go complete sequences without alignment? Because you can realign them as per your own program, as per your own choice later on, and we will do all these things in next session. So here I'm going to download this data, FASTA complete sequence. Just click here, and you can see here, so something is downloading. So let's see. uh, yeah, here you can see 2934. So once this data is downloaded, I will show you, and then this session will be over. For next session, how to treat this data and how to learn these things, for the next stage we will perform this in another lecture. That will be lecture 5. Okay, the download is complete, sequence dump, and I have already some other sequences, so you can see this 4. Okay, this is the here you can see, click here, and now these are sequences. Okay, so why some of the sequences seem very longer? Because I think there are some complete genomes also, possibly complete plasmid genome. So that's why I ask you to download the complete sequences. Now we have these sequences, and I will name it, let's say, NDM1 blast. And in part 5, we will explore these sequences further in offline sessions. Okay, thank you so much. If you are liking, uh, if you like this series, please thumbs up. And if you have any question, please ask. Thank you so much. Bye bye.